My name is Andrew Ferguson. I'm an associate professor at the Pritzker School of Molecular Engineering at the University of Chicago. And so thank you to the organizers for the opportunity to present as part of this virtual conference and the opportunity to tell you a little bit about our work in reconstructing all atom protein folding funnels from a low-dimensional experimental time series. So just a brief outline of the talk. And so the target is really to reconstruct single molecule free energy surfaces, SMFES for short, and molecular configurations from experimentally accessible observables. And so the manner in which we're going to do that is by processing one-dimensional time series of these experimental observables using a combination of ideas from dynamical systems theory, specifically Hawkins delay embeddings, manifold learning, which is a form of nonlinear dimensionality reduction, and some deep learning. And so I'll demonstrate the technique, I'll describe it in the context of a simple polymeric system, a C24 alkane chain, and then demonstrate its applications to more sophisticated and interesting systems such as Villain, BBA, trip cage, and Chignalin mini proteins. Okay, so first of all, what's a single molecule free energy surface? And so one can conceptualize a single molecule free energy surface as the following simple picture. So it's a high dimensional object, usually we can represent it in a few dimensions, 3, 4, 5, in which these low dimensional collective variables psi discriminate between the important thermodynamic and kinetic states of the molecule. We can then associate with each value of psi a value of the energy, U of psi, and entropy, S of psi, which together give us the free energy, F of psi. And so one can think about the altitude over this landscape as being the free energy. And then, of course, the free energy is related to the probability through the Boltzmann factor. So these objects, these single molecule free energy landscapes, have been incredibly valuable in helping us understand molecular behaviors, in particular the folding of proteins. And so this picture was introduced by Ken Dill and others in the late 1990s. And this statistical mechanical picture of a folding funnel in which unfolded proteins living at high free energies tumble down into low free energy native states really revolutionized our understanding of biomolecular folding. And it's also an important object for engineering and designing proteins to have particular folding characteristics, particular thermodynamic stabilities, particular kinetic folding pathways. So resolving these and recovering them from experimental and indeed computational data has proven to be a valuable endeavor in single molecule biophysics. So how does one recover a single molecule free energy surface from a simulation trajectory? Well, basically one runs a molecular simulation. So here's a simple example of a simulation of a C24 chain. It's living in water, but I've removed the water molecules for clarity. And one can conceive of this as being a high dimensional trajectory. Basically, we have n observations in some high m dimensional space of the Cartesian coordinates of all of the atoms of the chain. By running some dimensionality reduction, so we use diffusion maps, but you can use your favorite nonlinear dimensionality reduction algorithm, isomap, locally linear embedding, Hessian eigenmaps, whatever. One can then embed this into a lower dimensional space, y tilde, in which we have our n observations embedded into this k dimensional space. And so if you run the algorithm correctly, you can figure out what are the important collective variables discriminating the important thermodynamic and kinetic states of the molecule. And those are the collective variables in which to build your free energy surface, just by collecting probability histograms, taking the Boltzmann factor, the inverse of the Boltzmann factor to turn them into free energies. Okay, so how about experiments? So is it possible to recover something similar from experimental data? So first of all, what experimental data do we have? So typically experiments cannot resolve the positions of the atoms as a function of time, but we can resolve some coarse-grained observables. For example, the head-to-tail distance of the chain is something one can follow using some techniques I'll describe shortly. So what I'm going to claim is that one can do some processing to these one-dimensional or low-dimensional time traces in some experimental observables, and then combine those with some dimensionality reduction in order to find analogs of the single molecule free energy surface that one would have gotten if we'd had access to all of the molecular coordinates as a function of time, as we did in the simulation trajectories. Furthermore, I'm going to assert that these two free energy surfaces are actually related by a smooth and invertible transformation. So they are basically two different representations of the same object. And by learning this transformation, one can convert the landscape one gets from experiment into one that we would have gotten, the true landscape, from the molecular coordinate data. Okay, so to be concrete, I'm going to describe the approach in the context of the C24 alkane chain. It's a simple polymer chain. And so here is the target landscape. Here is the object that I recovered from running long molecular dynamic simulations off this chain. So one can see that we have two order parameters describing the configuration of this chain, NPC1 and NPC2. The terminology is not particularly important. And then the z-axis describes the free energy as a function of those two configurational order parameters. So one can see that down in the global free energy minimum, we have the all-trans configuration of the chain fully extended. Then there's a free energy barrier in which the chain forms a hairpin and curls up into this hydrophobically collapsed globule that lives in the local free energy minimum on the right-hand side. Okay, so this is the object that we would get if we have access to all of the molecular coordinates as a function of time. So can we reconstruct this from experimental data? And so I'm not going to talk about true experimental data, but it's sort of a synthetic, experimentally observable data set that I'm going to construct from the molecular simulation. So to make this relation concrete, I'm going to claim that we're going to be mimicking something that one could measure in the laboratory using a technique such as single molecule FRET. So FRET is fluorescence resonance energy transfer, also known as Forster resonance energy transfer, where one functionalizes a molecule with two optical probes. And so these fluorescent groups, here denoted by cyan fluorescent protein and yellow fluorescent protein on the right-hand side of the slide there, respond to light in a particular way. In particular, if one illuminates according to a wavelength of light of 440 nanometers, the cyan fluorescent probe absorbs that and then re-emits at 480. If the cyan fluorescent protein is in sufficiently close proximity to the yellow fluorescent protein, there's a non-radiative energy transfer from the cyan to the yellow probe, and then an emission of light at 545 nanometers. So by irradiating at 440 and then measuring at 545, one can then compute through a calibration curve how proximate these two probes are. And then by doing so, basically we've devised a molecular yardstick by which one can measure the distance between these two probes and then something like a distance in a molecule as a function of time. So by adding these probes to, say, the head or the tail of the molecule, one can follow the extent of the molecule as a function of time. This is going to be our experimentally observable time trace and a single coarse-grained observable. So the question becomes, can we reconstruct the free energy landscape from that one-dimensional time trace? If we had the landscape, we could reconstruct the FRET curve because we're going from a high-dimensional object to a low-dimensional object. There's no loss of information. One knows the configuration of the molecule and then can predict the FRET efficiency. The question is, can we go backwards? So from this experimental data, can we reconstruct this multi-dimensional folding funnel? So in other words, can we infer the geometry and the topology of this folding funnel, this single molecule free energy landscape, knowing only the time evolution of a single coarse-grained system observable? So the answer is yes, up to some important restrictions. Okay, so the thing that we're going to use, the mathematical theorem we're going to use to perform this calculation is known as Hawkins delay embedding theorem. So the theorem is described in the colored boxes on the left. Let me just talk you through the schematic diagram on the right. So in the upper panel there, in the space S, this is the Cartesian state space of our molecule. The molecule lives on a low-dimensional manifold in that space M. So these are the collective degrees of freedom that we found by applying diffusion maps, the nonlinear dimensionality reduction, to our Cartesian coordinate trajectory. And so upon that manifold, we construct our free energy surface. And so the arrow on that manifold is denoting the molecule moving around on the low-dimensional manifold in the high-dimensional phase space. Okay, so then just by collecting probability histograms over that manifold, we can turn them into free energies and construct our folding funnel, our single molecule free energy surface. So we don't have access to the Cartesian coordinates of the molecule in an experimental measurement. What do we have access to? So maybe we have access to one experimental observable, say the head-to-tail distance computed by FRET, as described on the last slide. So that readout is denoted as V on the right-hand side of that plot, and that provides us this one-dimensional time series, V as a function of t. Okay, so this is the object we're going to operate on using Hawkins delay embedding theorem. So Hawkins delay embedding theorem says, do the following, which might seem slightly strange, thing. Take that one-dimensional time trace and explode it into a vector space. So in the top of the blue box there is the delay embedding. We take the instantaneous measurement of the head-to-tail distance at time t, v of t, and we match it up with time-delayed measurements of the head-to-tail distance, v at t minus tau, t minus 2 tau, t minus 3 tau, all the way to t minus 2n tau. So now we have a vectorial representation of the head-to-tail distance at a number of points in time. So we can make that transformation, and that takes us down to the bottom diagram on the right-hand side, which projects the one-dimensional time trace into this 2n plus one-dimensional vector space. Hawkins' theorem then asserts the following remarkable thing. There exists within that 2n plus 1 delay embedding space a manifold phi of m, which is a smooth and invertible transformation of the manifold m at the top of the plot that we would have gotten if we'd had access to all of the Cartesian coordinates as a function of time. So what is that telling us? It's telling us that we can construct an image of that manifold that we require from the experimental time series by performing this Hawkins delay embedding. The smooth and invertibility is important because it means that the manifold is stretched and squashed relative to the true manifold, but it cannot be torn or stitched back together, meaning the topology of the structure is preserved. So this is sort of the no free lunch part of the analysis, that we can reconstruct the topology of the manifold. We know that we're going to get the metastable states correct and the connectivities between them, but we may screw up the heights of the free energy barriers or the depths of the free energy wells because there's an uncontrolled stretching and squashing of the manifold under the action of the delay embedding operator. Okay, so let me show this in practice to make these ideas very concrete. So here is our molecular dynamics simulation of the C24 chain moving around in water. Again, the solvent's removed for clarity. But now we're going to claim that we don't have access to the molecular simulation. All we know is the head-to-tail distance of the molecule as a function of time. So this is a synthetic fret time trace. And so, of course, it's not a true experimental time series because we have sort of infinite resolution at infinitesimal time resolution. But we are measuring observable that in principle is accessible to experiments. So given this sort of idealized time trace of the head-to-tail distance, can we reconstruct the free energy landscape, the single molecule free energy landscape, from this data by applying Hawkins delay embedding theorem? So the answer is yes. So here is the manifold that we've reconstructed. So what have we done here to recap? We've taken our head-to-tail time series, we've exploded it into this high-dimensional vector space using Hawkins delay embedding theorem. We've computed the appropriate time delay, this tau between successive measurements, and the number of measurements we need, n, using some internal consistency checks, and that's given us an explosion of our head-to-tail time series into this high-dimensional vector space. We then run nonlinear dimensionality reduction, exactly the same algorithm as we ran before on the molecular simulation trajectory, to find the low-dimensional manifold that's latent within that space. So here it is. It's parameterized by these variables npc1 star and npc2 star, and then by collecting the probability histograms of the data over that manifold, we can construct the free energy through the Boltzmann factor. So the z-axis is the dimensionless free energy constructed in that manner. So just by visual inspection, one can see that the free energy landscape that we have recovered looks visually very similar to that we got from analyzing the all-atom simulation trajectory. In particular, you can see that the global free energy minimum corresponding to the all-trans state exists there on the left-hand side. There's a free energy barrier corresponding to hydrophobic collapse of the chain, and then curling up into this helical hydrophobically stabilized configuration on the right. So Hawkins' theorem asserts that these two objects are diffeomorphic. They are smooth and invertible transformations of one another. But of course, we would like to verify that numerically. So basically, here's the recap. We have our simulation trajectory. We've run nonlinear dimensionality reduction over the Cartesian coordinates to learn the free energy surface from the Cartesian simulation trajectory. So that's the top line. On the bottom line, we've said that all we have access to is the head-to-tail distance between the leading and the trailing bead in the molecule. We've run Hawkins delay embeddings plus nonlinear dimensionality reduction using diffusion maps to give us the free energy landscape on the lower right of the slide. And then Hawkins' theorem has asserted that these two things are smooth and invertible transformations of one another. So how do we know that this really is the case? And so the inverse function theorem tells us that in order for two manifolds to be invertible, we need to simply compute the Jacobian of the transformation between them and check that the determinant doesn't change sign. So in the same way as you convert from Cartesian to polar coordinates, we have a determinant of that transformation. We're just computing numerically what is the determinant of the transformation between npc1 and npc2 in the original free energy surface, and npc1 star and npc2 star that we discovered from the head-to-tail Hawkins delay embeddings. So on the bottom left there, I'm showing basically a plot of the free energy landscape parameterized by MPC1 and MPC2, and I'm just coloring each point by the Jacobian determinant over the manifold. And so what one can observe is that the Jacobian determinant is indeed single-signed. It's always positive everywhere on the manifold. It's perhaps more apparent by plotting it slightly differently and just plotting the value of the Jacobian determinant as a function of time. So on the right-hand side there, you can see that Jacobian determinant is always greater than 0. It varies over around an order of magnitude, but it never passes through 0, meaning that indeed these manifolds are invertible. So importantly, the Jacobian determinant is not, although it's single-signed, it's not single-valued. If it were single-valued, that would mean that the Jacobian determinant between one manifold and the other is basically an identity. The manifolds are the same. The fact that it changes over the manifold is telling us that we're differentially stretching and squashing it at different locations over the manifold, meaning that we cannot trust the barrier heights or the well depths because we don't know how much they've been stretched or squashed. We can trust, however, the location of the metastable minima and the transition paths connecting them, because Hawkins' theorem tells us that the manifold cannot be ripped and cannot be stitched back together. So let me make that a little bit clearer in the context of an application to a couple more interesting systems, fast-folding proteins. Okay, so this is a small 35 amino acid protein known as villain headpiece. We conducted all-atom molecular dynamics simulations off this and also appealed to some long molecular dynamics simulations conducted by D. Shaw Research. So this is the reference for those trajectories in the bottom of the slide there. We took those trajectories, we analyzed them using diffusion maps, and constructed the low-dimensional single-molecule free energy landscape in the blue box. So this is parameterized by 2 collective variables that we discovered by our nonlinear dimensionality reduction, and then coming out of the plane of the slide and through the color map is the free energy. So you can see that there's a native structure for the protein, denoted by A. The molecular structure is in the bottom right. And then there's the large unfolded ensemble, B, is one representative example from that. So we then did exactly the same trick as we did for the C24 chain. We said, well, let's pretend the only thing we know about this molecule is its head-to-tail distance. Let's run Hawkins delay embeddings, let's run diffusion maps, and let's get the free energy landscape that way. So that is the free energy landscape in the red box there. So you can see, again, visually, there's very close correspondence between the two landscapes. Of course, there's a 90-degree turn here, but again, we can sort of verify by looking at the Jacobian determinant that the manifolds really are smooth and invertible transformations of one another. So in the bottom left there is the Jacobian determinant of the transformation as a function of time over these 120 microsecond simulation trajectories. Again, you can see that it is single-signed. It is not single-valued, but it only varies over about an order of magnitude. So I said that this is important because it discriminates how much stretching and squashing we do over different parts of the manifold. The fact that the Jacobian determinant tells us the degree of stretching and squashing only varies over a factor of 10 means that we don't stretch or squash too differentially, meaning that we can trust the barrier heights and the well depths to within a certain magnitude. And actually, by some subsequent calculations we've done, if the Jacobian determinant doesn't vary over more than an order of magnitude, the barrier heights and the well depths are correct within a few kTs. So this is good news. This tells us, at least for this particular case, we can trust the free energy landscape we get from the delay embedding up to order a few kTs. Okay, let me show you a second example for another fast-folding protein. This one is trip cage. It's a 20 amino acid protein. So again, we did exactly the same thing. We took the long D-shot simulation trajectories, in this case 208 microseconds. In the blue box is the all-atom free energy landscape we got from analyzing the simulation trajectories, the Cartesian coordinates as a function of time with diffusion maps. Again, we find in this case that we need approximately 2 collective variables, psi2 and psi3, to describe the configurational state of the protein, and then the free energy is computed by probability histograms and is shown by the heat map. So in this case, it's a little bit more of an interesting folder because we have A, which is the native state, we have B, which is a partially folded intermediate, and then we have a bottleneck region that takes us out into this large unfolded ensemble. And so we've just extracted one member of that ensemble, C, and all of those structures are represented in the bottom right. We then do exactly the same trick. We take the head-to-tail distance as the only observable we have access to. We apply Hawkins delay embeddings, we apply diffusion maps, and then we get the free energy landscape in the red box there. And again, you can see that there's very close visual correspondence. We map out the native state, the partially folded state, the unfolded ensemble correctly, and the Jacobian determinant is single sign and varies only over an order of magnitude. Again, telling us that we can trust the barrier heights and the well depths up to a few kTs. Okay, so how might one use this in practice? And so if one has access to long molecular simulation trajectories, one can do all of the protocol I described and calibrate the transformation between the landscapes. So what do I mean? One can run diffusion maps on the molecular simulation trajectory, get the all-atom single molecule free energy landscape in the blue box. One can analyze the head-to-tail distance as a function of time, run Hawkins delay embeddings and diffusion maps, get the free energy landscape in the red box, and then one can calibrate the transformation between those two things, denoted by phi and phi inverse. One can do this by simple interpolation, by some sort of grid method, or potentially some sort of artificial neural network. We've used all of the above. Depending on the system, one can work better than others. Neural networks appear to be the most powerful and the most convenient way of sort of representing that transformation, but any way of representing that function is completely adequate. So then what about deploying it in reality? So if you have a calibrated version of this embedding, then one can then analyze new experimental data, new head-to-tail data, pass it through the bottom pathway, run it through the artificial neural network to map it to the true free energy landscape in the blue box. Okay, so this all sounds pretty good. It provides a way in principle, so again, we haven't demonstrated this in true experimental data, but at least in principle for idealized one-dimensional time traces of experimental observables, one can do this. One can find the single molecule free energy landscape exactly through this transformation. There is one thing missing, so perhaps we would also like to take this head-to-tail time series and try and find not only the free energy landscape, but the molecular configurations over that free energy landscape. So is it possible to also do that? The answer is yes, and one way to do that is to train some deep neural networks to map us from the Hawkins delay embeddings of the head-to-tail time series back to the molecular configurational space. So let me just close with a couple of examples showing that this is indeed possible. This is relatively new work. But let me show you for, first of all, the C24 chain. So what did we do? We took the head-to-tail time series, which I'm going to show you projected in just a second, and trained a neural network to turn that into the molecular structure. And so we did that via the Hawkins delay embedding. So here it is. So I'm projecting simultaneously the head-to-tail time series as a function of time. Then we pass that through the neural network, and in the bottom right, I'm projecting simultaneously the true molecular simulation trajectory configuration in yellow, and the one that we reconstruct from the neural network in blue. And so you can see visually there's actually very good correspondence between these two things. We do make some mistakes because there is some information loss, and so it's not a perfect reconstruction, but quantitatively, the root mean squared deviation between the atoms of the true molecular trajectory and the reconstruction is lower than 1 nanometer. So that was a polymer chain. How about a protein? So here's an example for chignalin mini protein. It's a 10 amino acid protein. It folds into a beta hairpin. We did exactly the same tricks, and so here is the head-to-tail time series, and on the bottom right is the molecular dynamics trajectory, and then the reconstruction in yellow and blue respectively. And again, we can see that from the head-to-tail time series, passed through the Hawkins delay embedding, through the trained neural network, one can reconstruct the molecular configuration of the chignalin backbone to very high fidelity, again to just over 1 nanometer RMSD, which is actually pretty good. So with that, I'd just like to close. So I'd like to thank the folks that did the work. And so I did this work in conjunction with one of my ex-graduate students, Dr. Jiang Wang, Wei Chen, Max Topol, and one of my old postdocs, Dr. Haitham Sidki. And then funding from this work came from the NSF through DMS 1841810, from MOLSI through ACI 1547580, and through the MICOM Center at Argonne National Lab, supported by the US Department of Energy. So with that, I'm happy to answer any questions you might have, and thank you again to the organizers for putting together this virtual conference and for the opportunity to speak to you today.